All right, this is going to be a brief overview of some Mendelian genetics that's a little bit past just dominant over recessive. When we're looking at Mendelian genetics, we can kind of break it down into two separate categories. Genes that are in your autosomes, which are your first 22, right? Chromosome number 1 through 22, because humans have 23 chromosomes. And then that last 23rd pair, which is your sex chromosomes. They're either your X if you're female and your XY if you're a male. So there's this last pair that is non-homologous. So if you look at a human karyotype, this is chromosome pair number 1, this is chromosome pair number 2 and 3, 4 and 5. This last pair right here is our sex chromosomes. So they're the last one that unless you're a female, you could possibly not have them in a pair. And so this would be chromosome number 1, and there's a gene right there. These are homologous, because they have the same genes. You can notice that, look, they're the same length and they have the same banding pattern because the DNA is wound up densely in the dark parts. And so you can actually match them up. You can see that there's patterns when you look at this karyotype. So this would be like taking a snapshot picture of a cell as it's going through mitosis, and the DNA is all coiled up in its chromosomes, and we just line them up right next to their chromosome pair. So all of these right here are what we call our autosomes. And we typically have a pair. So if I inherited one, let's say big A from my dad, and the other one is different, I got big A, little a. But over here, let's see, there's another gene that controls somebody else. Let's say I have one version right here, little a and little a. Well, I'll just call that a little b and a little b, right? Over here, so we have a big Y and a big Y. So this is our genome. But over here, the sex chromosomes, the reason why we point them out as something different is because males inherit these genes differently. And now every other gene in our autosomes, we get them in a pair, and we don't know, is one dominant over the other? Is it co-dominant? Is it incompletely dominant? But with males, we only get one version. So we're only going to get either big A or little a. And that's what it is. That's what's going to be represented, because that's the only instructions that we have to produce that protein. But ladies, you get a set of them, so they act just like the autosomes. Many human genetic disorders are on our autosomes. This is an autosomal dominant disorder, meaning that if an individual has just one version of this disorder, then they have it. This is called limb mammary disease, which causes a deformation of limbs. And so this individual and these two genotypes would have this condition. So most of us are little a. And so what this highlights is that since we know that this is a rare condition, so anybody with these two genotypes would have limb mammary disease. So why is it that 99.9% of all human beings have little a, even though it's recessive? Because we know that recessive doesn't mean less frequent in the population. It just means that in the presence of both, one of them shows up. But since having this is bad for your reproductive fitness, any individual with either one of these two genotypes are going to die out before they pass on their big A gene. And so the only ones that do typically show up in the population are the little a's. So we typically see diseases as being a recessive, right, hidden with what we call a carrier. And so with this individual, we have the big C and little c. They carry the disease, which is this allele right here, that's being masked by the dominant C. And so they carry it, but they don't have it. So the only way to have it show up is two carriers and then recombine, like is right here, the little c allele. An example of this is cystic fibrosis. And so if you have cystic fibrosis, right, you could be a carrier for it. You can cross with another carrier. You do the Punnett square. Big C, little c. Big C, little c. 25% will have big C. They're full-blown, don't have it. Well, 50% are big C, little c. We call these individuals right here carriers. And 25% will have the disease. So that's what this shows. This is a pedigree. The very first row, Roman numeral 1, that's the first generation. That's grandpa. Right, this is a male. That's a symbol for male because males are square. And ladies are kind of curvy, so they're shown as a circle. And they're half because one is big A and the other one's little a, and this one's big A and this is little a. They don't have the condition, right? So this is recessive. And so this individual right here, right, these guys cross. That's what the line across means. That means they're sexually reproducing. And the line down means they have children and they connect to the oldest one, which here is the oldest female. The other one down is the second oldest, which is a male. This one is not a daughter of these two parents. She is the wife or breeding partner with the second oldest child. That's how this works. This is the second generation. So this is the third generation. So what is the relationship of this individual right here and let's say this individual, that would be like the father-in-law. This individual right here, right? This individual right here, and this individual would be like cousins because they have, this is her father and this is her father's sister. This is a dominant allele. Right? This, let's say this right here is dominant. This would be like the big A allele. This is the little a. And they're less common. This is achondroplasia or dwarfism. And so all you would need is one of the version of these alleles in order to have this condition. Huntington's disease is an example of an autosomal dominant condition. So this individual right here, who's little a, does not have it. This individual is either big A, or is big A, little a. We don't know because it's dominant. What we do know is everybody who doesn't have the condition is recessive because it's an autosomal dominant. So anybody who has a condition has at least one of the dominant alleles. We don't know unless we look at their parents. And then that'll tell us whether or not that they're going to have a little a. Well, if this is a dominant individual, right, and we know that everybody who doesn't have the condition is recessive, and they can produce a little a, then we know that they have to be heterozygous because they have to contribute at least one of the little a's to their offspring. Now, not every gene is autosomal. Some of them exist on our sex chromosomes. This is the Y chromosome, not because of its shape. It's just called that. This is the X chromosome, not because of its shape. It's just called that. Okay. As you can see, the Y is smaller. It has less genes, but this is all the DNA, all the U chromatin coiled up really tight. And this is one, and this is the sister chromatid attached to it. And there's the centromere right there. There's the centromere. So that's a bunch of DNA coiled up and then attached to its sister chromatid right now. And so this is a male. So none of the genes on this are existing on this. They're different genes. And so males only have one copy of their X. Sex-linked genes, like color blindness, are in males. We only have one copy. So whatever it is that we get, that's what we're going to be. So this is the story of my family. My dad is an unaffected father. So he has an X. We're going to call that normal. So we're going to write this over here. Big N, normal vision. And then little n is going to be color blind. I'm going to abbreviate that with CB. My mom's dad, my grandpa Joe, he had an X, he had a Y, and his X had the little n on it. Gave that to my mom right there. My mom got the broken X from my grandpa Joe. He was colorblind. Okay? So the way that we draw these Punnett squares, right, is that we first establish female, doesn't matter where you put them, and then male, X and Y. And since these sex-linked traits are for the most part on the X chromosome, there are genes on the Y chromosome, you put it as a superscript in the upper right-hand corner of the X, kind of like you're doing algebra, you know, X squared X. And so what happens is my dad, right, he is normal. So we put a big N right here in the upper hand corner of the X. We put nothing on the Y ever because this gene doesn't live on the Y chromosome. My mother is a carrier, so on one of her X's, she has a big N. The other one, she has the colorblind. So she herself has normal vision because colorblind is recessive, and she has this other X to kind of bail her out. And so when my mother and father had children, they had 4, right? Me and my brothers and sisters. What were the chances when they had their first child that the child was going to be colorblind? Well, let's do the Punnett square, because what a Punnett square shows is not what their children would be, but the probability in the outcome of that, of their children and their offspring. And so this is one possible combination of my parents' gametes. First, we look at the sex chromosomes, 2 X's, so that's female, right? So this is a female who is normal. This is another 2 X's, also female. One has the big N, one has the colorblind. So this is a female who's a carrier. Okay? This combination, X normal with a Y, this is a male, and C's normal. That's my brother Paul right there. Oops. Pot. That's Paul. And this is an X, N, and a Y. And that's me and my brother Mark, who's colorblind, and me, John. Now my sister Kristen is up here. She's either a carrier or she is full-blown, does not have it at all. We'd have to wait until she has children and know what the phenotype of her husband, if she gets married, has. And so even the chances of my parents having a colorblind child is only 25%, yet 50% of their children are colorblind. It's because a Punnett square doesn't dictate the actual outcome, it's just the probability. And so that's what happens because there's no second copy. So if you look at how does a female become colorblind then? This is really, really rare. Well, dad has to, at the very least, be colorblind. And mom, at the very least, be a carrier. So this is, we'll give her normal. So it'd be like my mom. She remarried a colorblind male, and then you do the Punnett square. What you'll see is this is my mother, who's a carrier, and then the colorblind male, who she's cheating on my father with. There you go. So we've got female, carrier, and right here, this is where you're going to get your female colorblind. Half of the males, right, are going to be colorblind. That's going to be a normal, and the other half are going to be colorblind. Now, it doesn't mean that all sex-linked genes are on the X chromosome. What about the Y? There are some genes on the Y chromosome. So you have the X chromosome and the Y chromosome, called these holindritic genes. Is it possible for a female to inherit a holindritic trait? This is hairy ears or hypertrichosis, which is on the Y chromosome. So if you have this X chromosome right here, and then the shorter Y chromosome right here, this male, that gene is located on the Y. It is impossible for female to have this gene since females have two X chromosomes. They never have a Y. They'll never have hypertrichosis. There is this special condition where the X chromosomes become kind of turned off or inactive, and that actually causes these calico cats, sometimes known as tortoise cats. And so a female has in her cells the X chromosome, let's say for black, there's black, right? And then there's orange. And so if they have two blacks, one of these X coils up into what's called a bar body, and it becomes inactivated. So then that cell is going to turn out to be black. And so if in their cells they have two oranges, that cell coils up and becomes orange, right? So it doesn't matter if they have black, black, orange, it doesn't matter. But if they have black and orange, right? Some of the cells, this chromosome turns off and black will show up. And some of the cells, this chromosome kind of turns off and deactivates and orange will show up. And then in some of the other hairs, you just see white. This is called X chromosome inactivation. Now, when they're spotted or tortoised, then they're heterozygous. They have one of each. Now, you could never see a male who has these spots because they would have to have two X chromosomes, one black and one orange. See, males are either black, and then they have the Y. A male is a female in here. She can be black. She can be, tortoise, right? Or she can be orange. But a male can either only be black, only be orange.